Hello, everyone. Welcome to our Avocado Virtual Field Day. I am Tim Spann, representing the California Avocado Commission. Thank you for joining us. I will say good evening, good afternoon, and good morning. We are covering a 17-hour span of time zones from the East Coast of the US to the East Coast of Australia. So we have people at all different times of day, and we appreciate everybody being able to participate with us, regardless of what time it is, wherever you are. So today we're going to be talking about Avocado Branch Canker. And I will just share with you a little bit of information and background on Avocado Branch Canker. Previously, this was known as Dothiorella canker. Some growers called it Dorothy. It's a ubiquitous group of pathogens that essentially can be found everywhere in all orchards, not just avocados, but other tree species. And these have been becoming more and more problematic in woody tree crops around the world over the last, say, decade or so. And so in avocados, we know this as Avocado Branch Canker. You can see the pictures there showing you what it looks like, the branch dieback symptoms in a tree. And then if you cut a cross section of a limb, you'll see that very characteristic V-shaped lesion that forms in the branch. And so in 2018, the California Avocado Commission started funding a project with our first speaker today to try and understand better the causal pathogens of this disease and to develop control strategies for it. And so the genesis for today's talk really kind of stemmed from two branches. One was we were discussing within the Avocado Commission, given the situation this year with COVID-19 and the lack of field days and grower outreach that we've been able to do, how could we reach out to the grower community and to have a virtual grower meeting? And at the same time, our two speakers today were having a discussion via email about the topic of today's meeting that I was looped in on. And I'm sure they'll never make that mistake again. And so when we started talking about having a grower meeting, I thought, you know, the discussion they were having was just so dynamic and so interesting. And it really, to me, it highlighted the thought process that researchers go through when they interact with each other and the importance of researchers interacting with each other. And so hopefully we can capture a little bit of that today in today's presentations. But that was sort of the genesis of having this meeting today. So our speakers today are Dr. Themis Michailides. And Themis, I have known Themis now, believe it or not, Themis, it's going on just at 20 years since I first met Themis when I started my PhD research at the Kearney Ag Center. Themis is a plant pathologist with the University of California, based at the Kearney Ag Center in the southern San Joaquin Valley. And he really is a world expert on Botrosphaeria, the group of pathogens that cause avocado branch canker. He's been working with this group of pathogens in nut crops in California for a number of years. And so when we decided to start exploring this in avocados, he was the most logical choice to approach to do this work for us. Our second speaker is coming to us from Australia today, and it's Dr. Liz Dann. She is a plant pathologist with the Center for Horticultural Science of the Queensland Alliance for Agriculture and Food Innovation. And she is an expert in tropical and subtropical crop diseases. If you've seen the avocado book, she wrote one of the chapters in there on Phytophthora, and I believe was co-author on a couple others. Highly regarded around the world for woody crops, specifically tropical and subtropical crop diseases. And she's been doing a lot of work in avocados in Australia. California growers, we need to be very fortunate that we don't have a lot of the issues that Australia has. They do have a number of pathogens and diseases that we don't have and we don't have to deal with, but there's a lot of similarities with what they are dealing with. So before I turn it over to Themis, just a couple of programming notes today. Both speakers will give their presentation, and then we will follow that with a moderated question and answer session. So if you look at the bottom of your screen, you should see a Q&A button, and you can type questions into that Q&A box throughout the presentations. Those will be monitored. We have Michelle Rothmeyer with us, who's been working with the commission. She does a lot of the writing and stuff for us in the background. You don't see her, but you certainly know her writing, and she is helping us out today to monitor those questions. And so then following both the presentations, we will go back and read those questions aloud and give the speakers a chance to answer them. And you can ask questions of myself, Themis, or Liz as we go along. Please we ask that you specify which one of us your question is addressed to as you type it in. If your question is answered after you post it, you can just give us a note to disregard that question, and we'll also be trying to monitor that as well. So with that, I will clear out of my screen, and I will turn it over to Themis Michailides, who will begin with his presentation on what he's been doing for the commission with Avocado Branch Canker. Themis?
Thank you very much, Tim, and good afternoon to everyone who is attending this webinar. So I chose this as the title photo, and these are not avocados. These are riparian trees because of the pathogens that cause the branch canker and dieback of avocado and the anthracnose. I will have some part of this talk on anthracnose because these pathogens occur at the same time in the same branches. I chose this picture because these pathogens live in these riparian areas. And the pathogens Botrytis, the members of Botrytis are not new to California. I had a circular from 1941 where they reported the Dothiorella rot, which is actually a decay of avocado fruit caused by Botrytis ribes, and also the Colototrichum, which caused the anthracnose of avocado. Another circular from the University of California dated 1935 was mentioned the Dothiorella canker. I couldn't find this in my notes, and so I do not have a picture of this. So these pathogens existed in California for years. Now, a century ago, there was a report, almost a century ago, a report of a disease of a walnut, which is called at that time Melaxuma. And in fact, that was a Botrytis canker, Botrytis disease caused again by the same pathogen, Botrytis ribes. This was dated in 1915. But we still have a lot of diseases caused by Botrytis avocados, and one particularly, the branch wilt, resembles the branch canker of avocado. These are walnuts, and these are caused by Neoscytallidium dimitiatum, which is a member of the Botrytis family. Very similar symptoms, entire branches are killed by this pathogen. We also have 19 more species, nine more species of Botrytis that cause a severe disease of walnuts, infect the walnuts, and from the walnuts invade the woody tissues, killing the spurs and the buds, thus reducing the yields. And you can see here also infecting the pruning wounds and causing cankers. We also have diseases by Botrytis in almonds, and these were reported back in 1950s. The band canker of almond, which can kill trees because it infects the trunks of the trees. But most of my studies and most of my experience is based on a particular disease, the Botrytis panicle and shoot blight of pistachio, which was discovered in 1984 in 2 isolated orchards in Northern California in Butte County. And it took about 12 to 14 years for this disease to spread throughout California and cause a major epidemic in the California pistachio industry. At that time, the epidemic was in 1998. We did not have any chemical control effective against this disease. And growers were desperate and they were cutting down pistachios and planting other crops like walnuts and almonds. We had cases where we could see by studying these fungi, these pathogens that cause panicle blight and shoot blight of pistachio, we found out that in young orchards, the inoculum was coming from riparian areas. These are elderberry along the Highway 5, and next to it was a pistachio or young orchard with no botryopsphere initially, but we started seeing the botryopsphere close to the highway, close to elderberry plants. The same thing in the almonds. Almond orchards isolated and surrounded by rice. Rice is not a host of botryopsphere. The inoculum was coming from the blackberries, where we found that the botryopsphere there were abundant in dead canes of the blackberries producing both water splash spores and airborne spores. I'll give you details later, and moving into the almond orchard and causing the branch canker, a very severe case in these almonds. But it's not very uncommon to find garden trees, trees along the roads, like sequoia trees, redwoods, cedar trees, covered with these blights. And all these blights are what? They are botryopsphere blights that eventually will kill the trees. You can see very severe botryopsphere infections of this blight of the cedar at the redwoods here. And we monitor some of these cases, and we found out that in 15 years, the fungus will not kill the tree by attacking the trunk, but it kills all the entire green tissue, and eventually the tree will die after 15 years. We have parks that are loaded with the botryopsphere diseases. This is a lot of ash trees with botryopsphere blights, and it's very common in the Central Valley to see this disease attacking all these trees. Now, over the years, we isolated the botryopsphere, members of botryopsphere from all these trees, including agricultural trees, forest trees, and we made a list that amounts to more than 50 hosts of botryopsphere. And this table actually includes fruit and nut trees and vines, ornamentals, and wild forest trees and bushes. So you can see from these that the botryopsphere fungi, the members of botryopsphere, have a very wide host range, attacking many trees in different families, so they're not very specific. When we checked the literature, we found out that the hosts, we had an exponential increase of the reporting hosts that were infected with botryopsphere. You can see this graph shown in the last decade, a very exponential increase of the first reports attacked by botryopsphere. And you can notice the studies on the control of the disease are very, very low, not very many. And this is an indication that these diseases that cause cankers are very difficult to control. Now, the botryopsphere species can cause different types of symptoms. So we can have leaf spots, fruit spots, diebags, perennial cankers, as I mentioned, in agriculture and forest trees and bushes and vines. And also, it's very, very common in any riparian areas, at least in California. These fungi act as saprophytes and also as parasites. Saprophytes means they can grow on dead tissues. If you have a branch that's on the ground, these fungi can colonize them and develop and reproduce. Parasites, they can attack directly green tissues. They also can be endophytes. The endophytes are these fungi infect the tissues, establish an infection with no symptoms, and they wait until the tissues become susceptible for the expression of the disease. And let's come now to the avocado branch canker and dieback. As Tim showed you, this is a major disease now. It's becoming more prevalent. You can see major branches killed, and that is direct loss of fruit, and also a reduction, loss of productive wood for the future crop. And if you examine closer these branches that are killed, you'll see definite cankers. In fact, in tissues below the margin of the cankers, you will see this discoloration here, which is almost like a reaction of the plant to this infection. We mentioned that the incidence has increased in recent years, and Tim mentioned that also. And the first report and support from the California Avocado Commission was with Dr. Scallen, who discovered that very many species of Botrys furiaceae can cause this canker. In 2017, the California Avocado Commission team called me, and he said that the industry is willing to support research, continue this research, and find out a solution and control, manage the disease. And so they funded a project that we started actually in 2018. And the objectives were, first of all, to establish a picture of how prevalent the disease was throughout the areas where avocados are grown in. And in the first year, we started with surveys of all avocado orchards, but as we learn more about the branch canker of avocado, we follow in the second year also sampling trees from nurseries and also from young orchards, because we had cases where young trees were dying. The second objective was to understand how this infection occurs, and also determine what are the most susceptible stages of infection. And we do this in our research in order to develop management strategies. More specific objectives we had were to determine the extent of Botryosphaeria and also the anthracnose presence in avocado groves, identify the most common and aggressive species. We knew that we were dealing with a group of fungi, both in Botryosphaeriaceae and Colletotrichum, but we wanted to find which of those are more aggressive, and we'll pay more attention to those in order to control. We wanted to determine the life cycle of these pathogens, also the disease cycle, how the disease is developing in the orchard, and what factors really affect the development of the disease. And finally, now we're starting with the disease control measures. You can see here that we know, and probably a lot of growers and the team and other groups there know that the sunburned tissues are very, very susceptible to branch canker. And you can see here, this is a sunburned tissue branch that is affected by Botryosphaeria, and you can see it's started dying. Also, pruning wounds can be infected, and pruning wounds can initiate cankers. On the left, on this side of this slide, you can see that a sunburned branch was infected and was killed. On the right is a sunburned branch that is not infected yet, but still is healthy. But eventually, this will be infected by Botrys furiaceae and will be killed. We also found out that we had infections, cankers that develop in graft unions, both in the nursery, even after they graft, immediately after they graft. And you can see here, these graftings have been killed. And also when they're in the pods, and also when in the field in the first or second year, we had death of young trees because of the graft union. Over the studies, we found that what parts of the trees can be infected. We can have here, I'm repeating myself a little bit here, pruning wounds can be infected, that will lead in cankers. Grafting wounds can be infected, they will kill the young trees. Wounds from hail, freeze, sunburn, all these wounds can be infected, will lead in cankers. Leaf tip burns burn due to drought, salt, or lack of organic matter. This will lead to leaf blight, and this can be infected by both fungi, the Botrytis and also the Coledotrichum. Cracks caused by drought or wind can be infected, and this is very characteristic of Botrytis. It can infect any wound that is created. Mechanical wounds can be also infected, leading to cankers. And the very unique characteristic of some of the species, they can infect with no symptoms. And these are the latent infections, and this happen mainly on the fruit and the leaves and the very young shoots, latent infections that remain with no symptoms until the tissues change physiology, and then we'll have the symptoms of the disease, fruit rot or leaf lesions, and even shoot blight. Now, we started the survey by surveying major areas in the areas where avocados are growing. We sample San Luis Obispo, orchards in San Luis Obispo, in Ventura, Riverside, and San Diego County. And from these samplings, we found out, you can see here, 5 fields were sampled. The black bars indicate the incidence of Botrytis in twigs and branches, and the red bar is the Coledotrichum. As I said, we isolate both these type of pathogens from twigs and branches, and this is very interesting for future research to see what the interaction is of these pathogens on avocado. So you can see here that the Botrytis are very predominant in most of the orchards in the San Diego County from twigs and branches. When we sample leaves in San Diego County, you can see that Coledotrichum is very high in some cases here, and in other cases here, Coledotrichum and Botrytis are very similar. So you can see that the Coledotrichum, the anthracnose pathogen, has a preference for the leaf infection, and this also indicates that sprays on the foliage will probably control this disease by reducing infections on the leaves and eventually infections on the fruit. Now, when we sample other tissues, and these other tissues can be even healthy leaves or healthy fruit, immature fruit, leaf scars and things like that, or we can find both fungi occurring there with sometimes with Botrytis high or the Coledotrichum high. But this is important because you can see that symptomless tissues can provide the same type of pathogens, but in lower levels. After we made a collection of isolates, we wanted to determine what groups of fungi we have. And morphologically, we noticed that they're very different, and also they produce different types of spores. And we do know, and we found also in avocado that these fungi produce both types of spores. They have the structures that we call pycnidia that produce the conidia, and these are spread by water. And the structures that produce the ascospores, that when they get wet, they shoot the ascospores in the air. In other words, this disease, the Botrytis canker, has both means to spread around and or even in long distances. And this stage here, the airborne stage, is never found on pistachio, although we have been examining this for almost more than 30 years. But it is very interesting. It is present in avocados. It is present on almonds and also on walnuts. Now, what happens when you have shoots with this pycnidia, these white structures here are the pycnidia. when they get wet, they use these spores as the toothpaste coming out from the toothpaste tube. And these spores with the water will dissolve and spread around. We also showed in other crops that insects can, these spores are sticky and insects can carry them around. The ascospores, you can see, and we found this in our leaves of avocado, produce the ascospores, and particularly this burn tip of the leaves has both structures, can have these pseudothecia, as we call them, producing the airborne spores. Now, of course, we wanted to see what species we have, and this is done with molecular techniques, and we found out that we're dealing with 11 different species in the Botryosphaeriaceae family. And so the next step, of course, is to determine which of these species are the most important and the most aggressive in avocado. This was not a surprise to us because when we did studies with walnut, pistachio, and almonds, we had 10 different species causing the disease on the walnuts, about 9 species causing the panicle blight on pistachio and almond. And also there are studies on the grapes where 12 different species of Botryosphaeriaceae kill the vines and cause cankers in the grapevines. A little bit about the anthracnose. The anthracnose also, it's a disease in avocado that is mainly affecting the fruit. We have infections of the fruit, and this has become more prevalent lately. And we see also in citrus in California, because the citrus industry has spread in the Central Valley, we see anthracnose occurring there and becoming prevalent as well. The anthracnose also is a very significant disease of tropical fruit. In a survey initially, in 2017, when we isolated the first time, 70% of the isolates were Botryosphaeria and 30% of Colototrichum. But lately in 2018 and 19, we see more, the incidence of Colototrichum has increased. Now, if you get, you put your avocados in the refrigerator and you forget about them, and then you examine them, you will see they're covered with lesions. And you can see gray lesions, gray color lesions, and this would be Botryosphaeria, and also pink lesions. And all those are the anthracnose lesions. And these are the latent infections. When you bought the fruit, you didn't see any symptoms, but when you let it mature, over mature, and decayed from all these latent infections that develop to extend and rot the fruit. In orchards, particularly in the coastal counties, we see this burn, the tip burn, and this tip burns from the salt can be colonized by Botryosphaeria and Colototrichum, and this is the saprophytic in a way infection of this fungi. However, once these are colonized, then the fungi can grow even into the green tissues and can cause leaf blight. Historically, the Colototrichum was known as Cleosporioides, but now this particular fungi, the Colototrichum, produce structures that we call cervuli, and these are producing spores that are water spread. And also in avocados, we found the perithecial stage of these producing the ascospores. Now, in Ventura, when we surveyed, we had orchards again with Botryosphaeria very high from different orchards here, and Colototrichum again in lower levels here in this particular case, about the same levels. But when you have leaves, in this case, you have more Colototrichum, in this case, about the same type. So it depends on the orchard, it depends on the inoculum, and this is from leaf lesions collected from these orchards. In San Luis Obispo, we had a case where the Colototrichum incidence was twice as much as the Botryosphaeria. So there are some orchards where the Colototrichum, the anthracnose pathogen, is much higher than the Botryopsphera. So when we did molecular studies, the complex of Coleotrichum gliosporioides separated in 5 different species. We're dealing with 5 different species. So we had 11 species of Botryopspheriaceae and 5 species of Coleotrichum. So then the next step is was to determine which of these pathogens species are most aggressive. And we do studies initially with detached shoots. And here I should thank Mary Larper, who are, allowed us to collect shoots from her orchard in Lincoln. And we inoculated this, and based on the size of the lesion, one can determine the aggressiveness of the pathogen. Here the results, you can see that some species are very aggressive. The higher bar here indicates the longest canker. These are Neophysicoccum. Again, they are members of the Botryopspheriaceae. The Lasiodiplodia is very aggressive, both on Cas and also on Gem, the same type of species. So these are the species that really cause major disease, more aggressive than the other species. And also the other thing that we have been doing is, and I prefer these, pathogenicity studies on live trees, and this is the avocados and podded trees. Avocados we inoculate, then we measure the lesion size on live trees, because this will give us what reaction the plants will do. And third, pathogenicity study, as well as inoculations throughout the season, are done in the pine tree ranch of avocado, down in Ventura, in Pomona, Ventura. And this is done both with spore suspensions and also with mycelia plugs. We use spore suspension with no wounding, and that's the natural way. These fungi infect the tissues, but also, as we said, when there's wounding, they can also infect, and this is where when we use mycelia plugs. By the results of these inoculations for 2 years now are not ready, but now we are collecting at the end of this year to find the differences in length size of the cankers and determine which is the stage of avocado that is more susceptible to infection, to create larger lesions, larger cankers. Briefly, I would say, based on the information we have so far, I know the time is going fast, the life cycle of these pathogens is as follows. We have all the inoculum in the orchard now in infected tissues, primary inoculum, and this will infect green tissues. Now, the green tissues, like the leaves, can produce a second generation of spores if we have secondary inoculum there. So we have a primary infection and then a secondary infection. And the primary infection of both primary infection spores and spores from the secondary infection from the leaves can infect the fruit. And also, if you have pruning spores, will infect pruning wounds, and then will close the cycle like this. Regarding the fruit set here, the flowers, we try to isolate from inflorescences here, but we could not isolate. We couldn't recover any isolates, but we probably need to do a little more plating and sampling of the inflorescences based on the information now we have from Elizabeth Dann from Australia. Now, lately, this is almonds, it's not avocados. We noticed that this, although the band canker disease caused by Botryopsphera was very sporadic, now we see very young orchards. This is a second leaf almond orchard with Botryopsphera throughout the orchard, very randomly widespread. And this indicated, suggested to us that, and this was an orchard with no obvious inoculum around the orchard. So this suggested to us that the disease came to this orchard from, with the trees. So we started doing studies on this, and at the same time, we thought about the avocados, because we did have cases where avocados were dying after grafting, and also, fortunately, not very many young trees were dying in some of those. And this was not the case in avocados, but in almonds it was like this. So this let us start a study to determine and identify if the Botryopsera fungi in the Colototrichum that we isolate from the young orchards or from the young trees are related to the sources. If there is a source of some coming from the nursery, or it is coming from the outside of the orchard. So so far as we know in almonds that a lot of latent infections come with the young trees, but in avocados so far we have some evidence that some coming with the trees, but we are not sure yet until we make the right correlations. But these studies with the molecular qPCR, which is a technique to identify the DNA, the quantity of the DNA of the species, will help us also develop a technique, a PCR technique that will help us to identify these fungi in healthy tissues, in tissues that do not show any symptoms, to see if there are latent infections. Both in almonds and prunes, we have evidence now with this technique to detect 5 different canker fungi with this technique. And this will help us then to focus our management strategies. Perhaps we have to start treating or paying attention to, after planting the trees in the orchard, how to keep the inoculum out of the orchards. And we are at the stage now to start in vitro, first with in vitro chemical efficacy studies, then to determine chemical assays with detached shoots, and finally to do field trials of fungicides, and also pay attention to kaolin. Kaolin is a whitewash. We know that sunburn is a major factor that affects the canker, and kaolin will be a major portion of these trials to see if, when we apply these, how well the branches are protected from infection by Botryopsera. We started some in vitro studies. This is Lasiodiplodia theobrome. This is not surprising because the same type of reaction inhibition we see with the same group of fungicides. You can see here the shorter bars are the ones that are shown in the fungicides that are more effective. We can see that Lasiodiplodia is inhibited very well from these fungicides that are 7-11 of these groups of fungicides, and also 7-11, 3-11, 3-9. And in comparison with the Botryopsera, you can see the same type of fungicides inhibit the Botryopsera, a less aggressive species, but also Botryopsera, a less aggressive species, is inhibited by the azoxystrobin, which the most aggressive Lasiodiplodia was not inhibited. So this is something that we started doing now, and then we'll continue with the treatment of shoots and determine among the fungicides which will be the candidates to be used in the field, including the kaolin, the whitewash of the branches. Some of the reasons here that explain the increase in the incidence are listed in this table here. First of all, as I showed you, these Botryopseraceae have very wide host range. And also, they act as secondary and primary. So that's another advantage they have. They increase the inoculum in riparian habitats. And we know that a lot of avocado groves are next to riparian areas, next to even forest. Increase of inoculum in non-clear forests. If you have a forest And the new practice now is not to clear this forest. And we see the effects now with the fires. That's where the inoculum will reside and invade the avocado groves. Many avocado groves are next to riparian areas. We know that. The disease can start. We have evidence now that the disease with the killing of some young trees, the disease can start at early age. And we have an inoculum buildup, and this is not noticed. It can be left if you see branches that are killed or trees that are killed. These have to be removed, because by removing them, you delay the appearance and development of the disease in the young orchard. And now we have growers that really push the trees to have more production, and that may cause a stress. And it is well known, and there are a lot of reports where drought stress and other type of stresses really affect the development of this disease. They predispose the trees to infection by Botrytis furiaceae fungi. We do have an experiment with water stress, and this is in progress at this time. So to conclude here, but based on these studies, we do know that the very sources of inoculum for the disease, the sources that produce primary inoculum, and this can include the branches, dead branches, green leaves, dead leaves, infected fruit, mummies that are left in the orchard, but also some of these tissues, the green leaves and the mummies, they provide two types of spores. The primary and the secondary can provide this type of spores. So, and also we are studying, and we know some of the factors that really affect the disease development in the orchard. And we are ready now to proceed with the control approaches. So I would like to thank you here. First of all of you who participated in attending this, but also I would like to thank the California Avocado Commission for the support of this project, the avocado growers who allow us to sample, Mary Larpea, Ben Faber, James Davis, Christopher Greer, all these help us to sample the orchards, and also graduate students that help in the laboratory and also in the field. And a special thanks go to Dee Vega, who helped us with the trials in the pine tree ranch. And I should not forget the nursery, the Brockhorn Nursery, who donated a lot of trees for the pathogenicity studies. And with this, I would like to end.
Okay, thank you, Themis. We appreciate that very much. As we get ready to turn it over to Liz Dann for her talk, I know there's a number of people who have joined us since we started, so I'll just remind everybody, if you go to the bottom of your screen, there's a Q&A button. You can click on that, and that will open a Q&A window, and you can type any questions you have into that window. And please specify whether you're addressing your question to Themis or Liz or myself, and then we will answer those collectively at the end during a moderated Q&A session. So with that, I will turn it over to Liz Dann, who is joining us from Australia. Liz.
Great. Well, thanks very much, Tim, and thanks very much, Themis, for your presentation. I think even though I'm just talking about panicle blight or flower dieback today, there's a very large number of similarities between your problems with branch canker and what we're seeing in Australia as well. So I think this is just the start of probably a long relationship of communications and sharing our research. So that's great. Okay, very happy. Thank you very much, Tim, for inviting me. It's great. One way that we can share some of this information over a great big ocean. So very happy to be talking to the Californian growers, and I think there might be a few Australians have hooked into the presentation as well. So I'll go ahead and share my screen now. Okay, that should have come up okay. Tim, that's all right, is it?
Not yet.
Not yet, okay. Oh, hang on, okay.
There we go. Yep.
Here we go, great. Okay, so this is an issue that really we've been seeing in the last couple of years, or it's in the last couple of years that we've become particularly aware of it. We're calling it panicle blight or flower dieback. Very briefly, I'll be talking about what it is, what does it look like. So I have quite a few photos of the symptoms. Is it new? I'll be answering that question. What causes it? And we're really just at the early stages of finding out all those causal organisms and factors. And very briefly, I'll be talking also about what are we doing about it in my industry-funded research project. So in the Childers Bundaberg, which is in sort of central Queensland, it's about 4 hours drive north of Brisbane, growers started reporting to my research team about October last year. So about this time last year, they started saying, oh, we're getting a lot of flowers are dying back and they're not setting any fruit. And of course, that obviously represents a big production loss. So my project was actually varied to accommodate some extra research activity to have a closer look at this. It seemed to be particularly prevalent in that Bundaberg Childers area, which is one of our major production areas in Australia. So the symptoms you can see, there's a browning and necrosis of the flowers themselves. And also for the indeterminate flower types, there's a shoot dieback as well. So really, really impacting those panicles. And over time, those panicles don't set fruit and they become skeletal within the canopy. Sometimes we see it as a wilt initially. So the actual inflorescence or the flower shoot can wilt before it goes into this necrotic phase. And for example, the picture in the middle is very typical of a dieback type symptom where a fungal infection is obviously progressing down the peduncle of the panicle. Again, the picture on the right shows what I'm calling a skeleton. So that's obviously died and been affected a long time ago. But as Themis mentioned, these sorts of structures are critical for inoculum for the following season. So we didn't only see this in the Bundaberg Childers area. I went to Western Australia for field trials in November last year, and because I was more aware of it, I was looking for it, and I also saw it in Manjimup, which is about 4 hours south of Perth. That's a very different growing region to Bundaberg Childers. So Bundaberg Childers is a typical subtropical production area. Manjimup in the southwest of Western Australia is very much a Mediterranean climate, so cold, wet winters and warm to hot, dry summers. So I saw it there as well, and also in New Zealand, which is probably a little bit more akin to the southwest WA type of growing climate. So again, you can see a severe dieback of those flower tissues and of the shoots arising, coming out of that indeterminate flower. The picture on the right shows some other damage to that peduncle, and that was probably caused by hailstorm. So again, as Themis mentioned, these type of wounds provide an ideal entry point for all sorts of pathogens, but probably particularly the Botryospherias. So then we did some work with some isolations from these grower samples, some that we collected and others that were sent in to us by the growers. What causes it? So we were able to isolate from symptomatic panicle material a lot of Alternaria, Cladosporium, Colototricums, Fusariums, Pestiloteuopsis, Diporthe, and that's the same as Phomopsis that Themis mentioned, and the Botryosphaerias. And as Themis has already introduced, we used to call that Dothriella, but we have new names for those things now. We were quite surprised that we weren't actually getting Botryosphaeria out at very high frequencies. So it was mostly Alternarias, Colototricums, and that has continued this year as well. And that was somewhat of a surprise to us, because normally in avocado pathology, we don't tend to associate Alternarias as causing a lot of leaf, fruit, or panicle blight type problems. But we think we need to look at it a lot more closely because of the frequencies with which we've been isolating that now. There was nothing that we've isolated that was particularly new or of biosecurity concern. So likely it's been in our orchards or our groves for a long time, but it's various factors have probably contributed to it becoming a lot worse in the last couple of years. Probably, as Themis has already mentioned, it's related to water quality and leaf burn. So in that Bundaberg-Childers area, the water quality is not good, and frequently the level of chloride is above 80 parts per million. And often it's double that. So that's probably another factor that's made the trees stressed. Also in those last couple of years, we have had a lot of dry weather, so drought type conditions, which is, even though the groves are irrigated, that type of drought stress is not good for trees. And the question is, does it progress to cause branch dieback? And the answer is probably yes, and they're probably linked, but we're still at the early stages, so I don't think we can really answer that question conclusively. In the top photo, that's a young tree you can see. It's probably only a couple of years old, and it's set no fruit. So this was actually northern New South Wales in December last year. So well after the flowering period, but the arrows there point to the flower shoots which have not set any fruit. And you can see also that it's obviously causing a little bit of branch canker in this case, because those leaves have also died back. So beyond the flower shoot, the leaves have been affected. There's a culture plate, so I'm glad I'm not the only pathologist that likes to put up photos of spores and colonies in Petri dishes. So we were getting a lot of Alternaria out of these types of panicle blight symptoms. As I mentioned, likely to be associated with the extended dry, droughty type periods. And I've just done a bit of a few graphs of last year's rainfall events leading up to the flowering season in the Bundaberg-Childers area. So in July, there was only about, July 2019, there was less than 10 millimetres. In August, again, there was maybe 12, 14 millimetres. September was extremely dry. Only a couple of millimetres in the whole of September, and that's when the peak flowering is. So peak flowering in that region is right now. So early mid-September is when flowers are out and are obviously vulnerable to infection during that time. And then it started to rain a little bit more in October. So while this isn't too surprising, because in Bundaberg-Childers it has a very summer dominant rainfall and winters are typically dry, I think coming out of very drought stress type conditions over a couple of years and then very, very dry July, August, September probably exacerbated the problem. And because it was dry, I think there weren't the leaching rainfalls. So even though growers typically with their low quality water and high chloride levels. They do leaching irrigations to wash the salt through beyond the root zone. There was no external rain or no natural rainfall, which would also have helped to leach the chloride through beyond the root zone. And Themis has already put up a photo of leaf burn. So we see a lot of the leaf burn symptom around Bundaberg Childers and other areas where the water quality is typically fairly poor. Already in 2020, the situation is quite different. So July had a rainfall of 35 mils, so that's well over an inch, and August as well, well over an inch. So that's a complete contrast to what we saw last year. So some grower and agronomist observations last year. As I mentioned, it was a big problem, particularly last year, although it had been seen in previous years. It seemed that Hass and Maluma were particularly affected, with no fruit at all in some blocks. The Shepherd is a green skin variety. It's a few weeks earlier than the Hass, and it was apparently not affected at all by this. And one agronomist noted that it was more severe in the determinate inflorescences and not such a problem in the indeterminate. And the predominant flowering type last year was the determinate type. So whether that's related or not, we're just not sure. It was a good observation to note down. More prevalent on younger trees, which is interesting. We're not sure why that is. It didn't seem to be related to plant growth regulator applications. Some orchards had used PGRs, others had not. And where azoxystrobin had been applied to try and get on top of it, that didn't show much success in slowing down the dieback or reducing the symptoms or helping those flowers and trees through the problem. So in some case, the new leaf flush arising out of the flowers continued to die back as well. And as I mentioned, in young plantings, there was shoot dieback, and we took some samples of these and have got some of those isolations done last year too. So whether all these things are interrelated, I suspect they probably are, but it was interesting that particularly last year there was an issue with this shoot dieback symptom, not obviously related to the panicles because these trees were far too young to be producing flowers, but there was an issue with these young trees not thriving over that winter period for us. So we went down to our favourite nursery and picked up some nursery trees, and even without inoculating, we were seeing these dieback in flowering nursery trees. So we're very good at killing things in the glasshouse, but we didn't even try to kill these. And they were just, they were happy to die themselves, unfortunately. I think this highlights that, yes, these infections are present in the nursery, even though when the trees leave the nursery, they're in fantastic health. It shows that they are probably still colonised. So those endophytic infections that Themis mentioned are probably already there, they're happening, and under stressed tree conditions and so on, they can be expressed. So you can see the two side-by-side photos as a tagged shoot, a flowering shoot, and over a period of 10 days, that dieback has extended even further. So obviously indicating fungal infection there. So then we started doing a little bit of a look-see what's happening in other crops, and in avocado we had only anecdotal reports of this flower dieback or panicle blight in avocado from California, and they were mostly conversations that I'd had with Mary Lou Apaya and also colleagues in Israel. But it was very difficult to try and find out who'd done the work on those. We know that in other crops, panicle blight is a big issue as well. And that's where Themis's name came up when we were doing the research and the literature searching. And as he mentioned, he's done a lot of work on this in pistachios. So my colleague, Femi Akinsanmi at UQ, has done some work in macadamia. And interestingly, one of the big fungal issues that he's found was Pestaloteuopsis. So another one was also botrytis, which is a little bit different. But in macadamia, his research has shown that the Botryospherias and others are also present and causing branch dieback issues. So I think it's analogous to what we're seeing in these other crops as well. This year, we're also seeing it. So there's no doubt that it's still present, even though the rainfall events are a little bit different. These are photos that growers and agronomists have sent me because they know that we're doing the work on this now. So already from August, September, we were seeing those same symptoms, not only in the Hass variety, but also in the earlier Shepherd variety. So then what are we doing about it? So it was important that we communicate this to industry. So I'm thanking you for the opportunity to include some of our Australian growers and industry members on this webinar. So it's important that we get samples from other growing regions and do those isolations as well, just so we get a bit of a handle on how widespread it is and what might be the key organisms or fungi in other growing regions. Earlier this year, Yuping Liang was a visiting scientist with my group. She came in from Taiwan about 2 weeks before everything went into shutdown. So she came at the end of February, and we're very fortunate to have her assisting us. And with Dr. Akila Prabhakaran, they've conducted these in vitro petri dish tests to show the relative efficacy of different fungicides. So we actually had 7 isolates. They were across Colototricum, Alternaria and Botryospheriae, those key groups of fungi. And we had 12 or 13 fungicides or biofungicides. So it was a massive, massive task. There are well over 2,000 petri dishes involved. And so each of those petri dishes has to be poured with different dilutions of the fungicide and then measured when those colonies started to grow. That's how we can determine which concentrations and which fungicides were going to be most effective against these fungi. So the bottom right, bottom left there, sorry, you can see that's a typical Colototricum, and the middle photo shows an Alternaria. Also lucky to attract Montana Hickey. She did a short research undergraduate project with us previously, so she wanted to come back to our lab for an honours project. So she started recently, and we've got her for probably not quite a year now. And her primary task, initially at least, is to review the literature more thoroughly than what I've been able to do so far. She'll be doing the isolations from the grower samples that we're hoping to get in this season, and she will be conducting pathogenicity tests in the glasshouse to inoculate with these various different fungi and try and replicate the symptoms. And that's just to determine how many of these fungi, which ones, and which ones might be more aggressive or cause the worst symptoms. So it's important to get that information as well. She's also very keen at looking at what are the environmental factors, for example, rainfall, humidity, temperature type data historically, and also from this year. So just as a way of linking what were the factors leading up to a severe problem last year, and we're not quite sure if we'll see it to the same extent this year. She'll also go through and do some molecular analyses just to confirm causal organisms and get the names right, basically. What else are we doing about it? Well, because our in vitro tests were extremely thorough, it came up with several candidates, and these are fungicides and biofungicides which have not previously been registered in avocado. So they cover 5 different chemical groups which are not currently registered in avocado. So Kayleen, who's actually sitting in the room with me, is very keen to look at the endophytic type infections, and because we have her experience, it was great to get a pre-spray sample of those this year. And then we'll also get a post-spray sample. So we've got the baseline from these tiny little match head size fruitlets you can see. And then we'll also be assessing what fungi we can maybe pull out from the similar sized or slightly larger piece size fruitlets after those first 2 flowering sprays have gone on. Normally in avocados, the recommendation is not to spray at flowering, but I've been asking growers why is that the case? And responses have been mixed. Some people say, well, it'll damage the flowers, or the bees won't like it, or the apiarists, the beekeepers, don't want anything sprayed in the orchards during flowering. But I think with some of these other chemistries, it really gives an opportunity to put some of these curative type fungicides on, and they're likely to be safer than possibly the ones that have been used in the past. One agronomist said to me, oh, well, I thought that the coppers dried out the flower, the stigma and anthers. So it'll be interesting to see, now that we have put these first 2 sprays on at flowering, actually what happens. So as I mentioned, both these field trials are in the Bundaberg-Childers area. One of the trials is in an orchard which was particularly hammered by panicle blight last year. The other one didn't have such a problem with panicle blight. We've got the 4 curative fungicides which are not currently registered, as well as copper, Serenade Opti and Amistar, and of course an untreated control which will go right through the season. We want to assess not only severity of the panicle blight and do our isolations, but also at the end of the season, have a look at yield, have any of our fungicide treatments through the season, and particularly at that early flowering stage, increased yield, and then also to take the sample of fruit and assess them for anthracnose and stem end rot. So in my trials, I religiously do that anyway, to see what effects any treatments have had on anthracnose and stem end rot severity. If we get some promising candidates for that, and I've spoken with the chemical companies involved, and they had input actually into which candidates I chose for the field trials. So they were candidates which were more likely to be progressed through to permit or registration with our APVMA, which is our fungicide and veterinary chemical regulator. So hopefully we might get some new information to support those additional registrations or permits. So thank you. That's a very brief overview of what we're doing and where we're up to. As I mentioned, we've really only started looking at this in the last year or so, but I have other members of my team who are looking at the branch dieback and also looking at pruning wound and hail fungicide dressings, which fungicides might be more effective under those simulated dieback and hail conditions. So there's a range of people I'd like to thank. They've been very supportive of my team's research, and of course my avocado pathology team at UQ. So thanks to everybody. And my project's funded through Hort Innovation, and the growers, avocado growers, pay an R&D levy. And so that levy gets used and that goes towards our R&D funding. So thank you, everybody.
Thanks, Liz. So I see that we have one question that has popped up for Themis, and there's another one for Liz. Michelle, do you want to go ahead and start with the one for Themis?
Yeah, the first question is, have you observed differences in the degree of damage caused by Botrosphaeria between Haas, Lamb Haas, and Gem and or other varieties?
There are differences, but we have not summarized them to present them to you, but they will be probably presented the report, the written report.
Okay. The next question is for Dr. Dann. In one of your earlier slides showing the skeletons, there was a dieback from the panicle towards the base of the shoot, but there was also a lateral shoot growing that seemed unaffected. Do you expect that lateral to become symptomatic too, like a progressing disease front?
Yeah, it's quite interesting actually with these smaller branches and twigs. Often the dieback is arrested at the node and it frequently doesn't progress beyond that, but sometimes it does. So I can't tell in that, I know the slide you're talking about, I can't gauge whether that would progress or not. It probably depends on health of the tree and environmental factors which may cause that fungal infection to progress, but often not. Often those skeletons are just limited to the panicle and it just stops at the node, which is interesting. Why does it stop there under some circumstances, and why does it potentially progress to blight that whole branch?
Okay, so again, feel free as we continue discussion here to post any questions you have using the Q&A feature at the bottom of your screen. I'll address this one and let both of you have a chance to answer. Given the prevalence of these pathogens, you know, in particularly in California, and I assume in Australia as well, we encourage growers to allow the leaf litter to accumulate and develop that mulch layer under the trees, to, you know, chip their prunings and leave them in the orchard. How detrimental is that? You know, and then particularly for us in California, we're putting that litter on the orchard floor, and then we're using micro sprinklers to irrigate, and so we're moistening it. So we're probably stimulating sporulation. So, you know, from an orchard sanitation standpoint, what could growers do? And is there any chance that as the material is on the orchard floor, will it begin the decay process, and how quickly would these pathogens die in that mulch and composting process that's occurring? So
I can respond to this, Tim.
Yeah.
This is very detrimental in avocado growth because both the airborne and the water splashed spores are present. This litter, I remember when we collected litter from an orchard, we can find there the pseudothecia or Botrosphaeria, and once these get wet, the spores will become airborne in the orchard. So it's very easy for the inoculum to spread around in this way. especially when you have a layer of litter leaves and even small branches with pycnidia and pseudothecia, providing all this inoculum coming from the ground, from the litter. Now, we show in the walnut orchards when we examine litter from debris from the huller after they harvested, about 70% were Botryopsera, a little twigs with Botryopsera, and about 20% were Phomopsis. But when they composted that material and we tried to isolate these pathogens, we got to 0. So I don't know how easy it will be to compost this material, collect and compost it, and then put it back as organic matter. But it is, especially for avocado orchards, seeding that material in the orchard is very detrimental. And in fact, I saw a grower was putting shreds from eucalyptus, and eucalyptus is an excellent host of Botryopsera. So.
Yeah, I can comment too, Tim. I'm completely conflicted on this because I also work with Phytophthora, and the healthiest roots under trees that I've seen have been in orchards that chip and have a really nice layer of leaf litter. So a very difficult one. While the whole orchard hygiene is important, and pruning out and removing these sources of inoculum, so the dieback branches and the skeletons, they are very valuable. And in avocado, the best mulch is avocado branch all chipped up. Perhaps if they're very well chipped up, that might be a way of reducing those inoculum sources. I think that taking them out and then composting them and bringing them back again is probably not going to be cost effective. So I think it's a case-by-case basis. Is the Phytophthora problem worse, or is the branch dieback panicle blight problem worse? Yeah, very difficult to answer that.
Yeah. Okay, I see we have a couple more questions that have been posted. Michelle?
Yep, this may be for both of you. So the comment is, so is no leaf matter then good under the trees? I think that's in response to the most recent question.
As I just mentioned, I love leaf litter. So I've got photos of avocado roots growing up into that lovely, moist, high organic matter layer that the fallen leaves have created. So I think I still prefer to have a good layer of leaf litter and organic matter rather than removing that valuable resource straight from the orchard.
If there was a way to spray something to decompose that litter, is that okay for the Phytophthora? Because that will be okay for the Botryopsera also. To if the litter is decomposed soon, then will not have a chance to spread spores around. I know sometimes the people spray ammonia. In some other states, they try that in leaf litter. To decompose the Perithecia venturia, but I don't know if the avocado leaves, because they are more leathery, in addition also the pistachio leaves are like that. I don't know how fast they will be decomposed and what will the cost will be by using a spray of ammonia in the orchard.
Yeah, potentially, yes. I think we probably do need to look at some sort of top dressing with something which could help increase the speed of breakdown. Obviously, we wouldn't want to put on anything that would also knock out the beneficial microbes, because they're the ones that are doing the breakdown. So perhaps there's a little bit more work to be done on that, on how we can keep this valuable prunings and natural leaf litter on the orchard floor, but perhaps to break it down a little bit more quickly so that it's not a source of inoculum for those infections. later on.
So that just made me think, Liz, would there be potentially, thinking about the life cycle of the pathogens, would there be a better time of year, particularly for California, where we have our very dry summers and unfortunately all too often very dry winters as well? Would there be a better time of year to prune and chip that material to give it a chance to break down before potential winter rains that would stimulate the sporulation?
Yeah, I think so. So I think it's looking at when the peak infection periods are and what are the conditions which cause the release of those spores, whether they're the asexual spores or the sexual spores, and then trying to time pruning and other management practices around that. So I think it's probably a case-by-case basis for the different regions, and yeah, really nutting it out a little bit further than we have already. I think that's going to be critical.
Well, for the for other crops, for instance, for walnuts, we compare pruning in the fall and pruning in the winter, and we found major differences in infection. And also, we recommend to possibly prune at a time when expected to have a drought. If you know exactly when that will happen, try to avoid a rainy season, or prune when it's foggy. Because those pruning wounds also can be infected. So it makes a big difference when you prune. I'm not sure in the avocados when the growers prune, what is the period of pruning, but probably it will be worthwhile to compare the two extremes and find the right time for pruning to reduce the infections.
So we have another question that relates to the mulching, and the question is, would it be better to collect the avocado leaf litter and clippings, but spread a different type of mulch without the bud infections? I think this is for either or both of you.
Yeah, if that can be done, potentially yes. I still prefer the idea of encouraging a more rapid breakdown, and that would still be my preferred option. But yes, if there's another source of material available, a lot of our growers have a hard time actually getting a woody mulch. So they often will get wheat stubble, straw out of broadacre production, and they'll spread that underneath their trees, or sometimes sugarcane mulch. So in Bundaberg Childers, they are growing a lot of sugarcane. So that is also a source of mulch. So yes, there are other things that people are putting on which are probably not hosts of these pathogens that we're talking about. So the levels of inoculum in those other types of mulch materials will be lower. So yeah, that is also an option. But perhaps they could go over the top of the natural leaf litter and keep the natural avocado leaf litter there in the first place. Big trees are almost self-mulching if everything's going right, because they do drop their leaves and they should be pruned. So they should not require any additional inputs of mulch once they're a nice, big, healthy, mature canopy. But if trees are getting smaller, and industries in California is no different, are looking at smaller trees for management and sorts of reasons, and young trees, they are not producing the same volume of their own natural mulch. So that's when alternative off-farm sources of mulch are brought in.
For avocado trees that do not have tip leaf burn, leaves, we don't find lesions of Botrytis or the Coledotrichum. So it is okay for the leaves to fall on the ground and decompose. But in orchards where we have leaf tip burn, these are the ones that are problematic, because they will have the both types of sporulation I will, in the majority, we see the leaves are healthy. And so the leaves is, I think the leaf litter is more safer than the prunings. And the prunings are the ones who will be decomposed the slowest. And the prunings are the ones which carry heavy loads of pycnidia or pseudothecia that will produce spores. So if there was a way for the growers, at least the prunings to collect them and chip them, compose them, and then use that as organic matter back in the orchard.
Okay, this next question is for Dr. Michailides. Can you talk a bit about how you will evaluate the salt interaction?
The self-interaction will be the interaction of the Botryopsera and the Colotochum. Is that the question? I believe that it is. So then this is done with control experiments in which we inoculate first the Colotochum and then the Botryopsera and vice versa, or simultaneously, then we'll compare the results of the infection. We have done similar studies with the interaction of Botryopsera and Phomopsis, and it makes a big difference which pathogen attacks the shoot first. So that's probably will be done in the orchards, in our experiments.
Themis, I think the question specifically wanted to know about an interaction with salt, salinity, but are you?
Oh, that was the question.
I don't think you're doing any trial.
No, at the present, we're studying the water stress, but that can be done also with a control experiment, irrigation experiment with different levels of salt to create the stress in the leaf burn. So.
Next question is for Dr. Span. With the damage from the heat wave we just had in California, should we expect to see more of the branch canker issues developing?
Absolutely. Yeah. As Themis alluded to throughout his talk, anything that causes physical damage, so sunburn, salinity, anything that gives an entry for these pathogens is going to allow them to explode and take off. And so we're going to have a lot of wounds on the trees following the heat wave. Those wounds are going to be can be infected, and hopefully we're going into the rainy season. So you're going to have a lot of spore activity in the orchard. So I think absolutely it's definitely going to be something to be on the lookout for as we go through the winter and into next spring. And then, look at those sanitation practices, get in there, prune out any of that dieback that you do find. And if you can get it out of the orchard, if not, chip it as small as you possibly can so it breaks down as quickly as possible.
One of my pictures, that orchard that was getting inoculum for elderberries, probably you noticed that there were garbage bags in the orchard. That was at a time when we did not have chemicals, and the grower was pruning the blighted shoots, putting them in those garbage bags and removing them from the orchard. And that was a very successful technique approach to reduce the disease and delay the disease in this orchard until we discover all these fungicides, effective fungicides against the Botryopsera of pistachio. And there's no concern about this any longer. The heat, the other thing that we have to remember that these fungi, the Botryopsera group of fungi, they prefer high temperatures. So they thrive under high temperatures. Particularly one of the species, Lacilodiplodia, and also the Neoscytalidium, it's heaven for them when it is really high temperature.
So the next question, maybe all three of you can answer it. It relates to high density planting. And it says, there is a movement toward high density planting. Do you think disease pressure will be even greater, especially with varieties like Gem and Lamhaus that tend to hold their fruit inside the canopy?
Yeah, definitely, no doubt about it. Anything that reduces the airflow through the canopy will certainly exacerbate disease issues. So not just the types of panicle blight, branch dieback, but also anthracnose and stem end rot in fruit. Often, if the density is so high, machinery access is going to be limited. So maybe it's not going to be possible to put on the fungicide sprays, protectants and curative fungicides that have gone on in the past. So I think we probably need to be quite careful about the move to high density if the management practices may suffer, or if fruit quality can potentially suffer. So I guess that's putting on my my fruit quality side of my project, anthracnose and stem end rot. I think we do need to be quite careful about the move to high density. But if it's managed and if access for sprays, if sprays can still get right through into the canopy to protect that fruit, then yes, there should be no problem.
I totally agree with this. There are examples where of dense walnut orchards. Growers are using hedging to prune. They create a layer of pruning wounds, and you can see Botrytis blight, Botrytis infection of pruning wounds up to the top of the canopies. In addition, the dense planting, in addition to what you said, Elizabeth, reducing the airflow, it will create, I'm not sure about avocado, but in other crops, the dense planting creates so much shade in the lower branches of trees that weaken the branches. And the weakness of the branch is a stress for the tree, and those branches are colonized saprophytically by Botrytis, covered, the entire branch is covered with pycnidia of Botrytis. They thrive on those branches, and this is what we recommend to walnut growers, to remove those out, because it's a high level of source of inoculum.
I agree with Liz and Themis. I don't have anything to add. I think they hit all the points I would have.
Okay. The next question. Has there been any work carried out on effect of the soil-borne beneficial organisms like Bacillus and Trichoderma on the pathogenic fungi and their spores, found, such as Calitrichum, Bactrospira, and Alternaria?
Years ago, we tried Serenade, which is Bacillus, against Botrytis with no success. But Serenade was registered the following year for controlling of Botrytis.
Yes, so we also did some work with field sprays with Serenade. It was a previous formulation to what is currently registered. So now in Australia, Serenade Opti is registered for foliar sprays, Serenade Prime for soil applications. I've not personally done the work with Serenade Opti, but it is one of the fungicides in my trials this season. So it'll be very interesting to see how that performs. Bayer do actually say, though, that it's not going to be a standalone and it's not going to probably be so successful under high disease pressure conditions for those fruit and foliar type conditions. As far as applying anything beneficial to soils, I'm all for having a very, very diverse and microbially very active soil community. So I think the best roots grow and thrive in soils which have a high organic matter to introduce natural microbial populations and diversity. And then once the roots are happy, the trees generally tend to be fairly happy as well, given the right management and water and nutrition and so on. So generally, but not always, there's a good relationship between tree health and what happens above ground as well. So all the management strategies need to come into play, not just what you might spray above ground, but also what happens below ground as well is crucial for orchard productivity and tree health.
I think, I agree with what Liz and Themis said, and I'm typically not a fan of, bugs in a bottle. Doing what you can to encourage the native soil bacteria and fungi, you're going to be far better off because those are adapted to your specific orchard. But thinking about it, going back to, what Themis brought up earlier, potentially looking at, something to help break down the leaf litter and the clippings and everything. Potentially, maybe from that standpoint, there might be something there that could work, not necessarily specifically against the pathogens, but just introducing some other fungi or bacteria that would help to break down that leaf litter and the clippings more quickly until you get that really good active mulch layer going that's encouraging and stimulating the natural native bacteria and fungi in the soil. Might be a potential thing to look at.
Excellent point. Mm-hmm.
I actually visited a mulching and composting operation, and they send truckloads of different types of compost and mulch all over the place. And they do inoculate their woody wood chip piles with a secret mix, I guess, of probably bacteria and fungi. And they claim that helps speed up the breakdown process. So I do think there probably is a role for looking at which of these bugs in a jug might be effective in making that breakdown process a bit more rapid.
Okay, well, I don't see any more questions on the Q&A. So I will thank you both again. Liz, thank you. Themis, thank you.
Thank you.
And to everybody who is still on, I failed to mention at the beginning, this entire session has been recorded and it will be posted to the Avocado Commission YouTube channel. So if you are a California grower or Australian grower, the California Avocado Commission has a bi-monthly email newsletter called the Green Sheet. And when it goes up on YouTube, we'll put a link to it in the Green Sheet. If you don't receive the Green Sheet, you can go to CaliforniaAvocadoGrowers.com, and there's a link there where you can sign up for the Green Sheet, so you'll be notified when that's posted, and then as well as any future meetings that we have. So with that, I thank our speakers one last time, and we will sign off. Thank you all very much for attending and participating.